So today we are going to discuss autoimmunity and what is autoimmune diseases. So now about peripheral tolerance. There are several backup mechanisms which occur in peripheral tissues to counteract the self-reactive T cells जो कि escape कर चुके हैं from the central tolerance. The first one is the ignorance. इसमें क्या होता है? The self-reactive T cells might never encounter the self-antigens which they recognize and therefore remains in a state of ignorance. That means वो कभी contact में आते ही नहीं है, encounter ही नहीं करते self antigens को जो कि normally वो recognize करते हैं, तो और basically stay in a state of ignorance, तो वो identify नहीं कर पाते हैं, तो इसीलिए there is we have immunological tolerance in our body. So second is the energy. Energy is unresponsive to the antigenic stimulus. So what happens here is basically the normally T cell जब activation होती है वो require करते हैं दो signals. एक होता है main signal, एक होता है co-stimulatory signal. तो energy में क्या होता है कि भाई जब एंटीजेनिक स्टिमुलस आता है तो मेन सिग्नल तो चलो इट इज कैरिड ऑन, लेकिन जो बेसिकली को स्टिमुलेटरी सिग्नल होता है ना वो ब्लॉक हो जाता है. तो नॉर्मली क्या होता है कि जो टी सेल एक्टिवेशन है उसका जो मेन सिग्नल है जो कि विच इज प्रोवाइडेड बाय एंटीजन मेजर हिस्टोकम्पैटिबिलिटी कॉम्प्लेक्स ऑफ एंटीजन प्रेजेंटिंग सेल्स, वो इंटरेक्ट करते हैं विद द टीसीआर, दैट इज टी सेल रिसेप्टर्स ऑन टी सेल. एंड एक को स्टिमुलेटरी सिग्नल होता है जिसमें एंटीजन प्रेजेंटिंग सेल्स में एक B7 मॉलिक्यूल्स प्रेजेंट होता है जो कि बाइंड करता है CD28 ऑफ द टी सेल्स. तो मेन सिग्नल तो कैरी ऑन एज सच ही रहता है, लेकिन जो को स्टिमुलेटरी सिग्नल होता है ना उसमें एंटीजन प्रेजेंटिंग सेल्स जो होते हैं ना वो B7 मॉलिक्यूल वो इंस्टेड ऑफ CD28 के अटैचमेंट के बदले में वो अटैच कर जाते हैं टू द CTLA4 molecules on T cells. So in this way the co-stimulatory signals get blocked and there is unresponsiveness to the antigenic stimulus. Now third is the phenotypic skewing. इसमें क्या होता है? तो self reactive T cells interacting with the antigen presenting cells which are present on the self antigen undergo activation, लेकिन वो non pathogenic cytokinins release करते हैं. Hence चाहे वो एक्टिवेट हो जाते हैं, hence they are activated, but वो autoimmune response नहीं generate कर सकते. Next is the apoptosis. Apoptosis by activation induced cell death. इसमें क्या होता है कि the activation of T cells जो होते हैं ना वो induce करते हैं upregulation of fast ligand जो कि subsequently interact करता है death receptor fast से, which leads to the apoptosis. So this is called as activation induced cell death. So we can only write कि जो self reactive T cells होते हैं वो क्या करते हैं, जब उनकी एक्टिवेशन हो जाती है टू द एंटीजेनिक स्टिमुलस तो ये अप रेगुलेशन ऑफ फास्ट लिगेंड करते हैं विच लीड्स टू द एपोप्टोसिस, राइट. सो नेक्स्ट इज द रेगुलेटरी टी सेल्स जिसको हम ट्रेग सेल्स भी कहते हैं. ट्रेग सेल्स जो होते हैं वो डाउन रेगुलेट कर देते हैं द सेल्फ रिएक्टिव टी सेल्स को थ्रू सिक्रीटिंग सर्टन साइटोकाइनेंस जो कि सेल्फ रिएक्टिव टी सेल्स को बेसिकली रेगुलेट करते हैं, उनको डाउन रेगुलेट कर देते हैं. और वो क्या रिलीज करते हैं? वो साइटोकाइनेंस रिलीज करते हैं जो है इंटरल्यूकिन 10 एंड ट्रांसफॉर्मिंग ग्रोथ फैक्टर बीटा. और ये बेसिकली by this the peripheral tolerance occurs. Next is the dendritic cells. Dendritic cells में क्या होता है कि ये डाउन रेगुलेट करते हैं द एक्सप्रेशन मॉलिक्यूल्स ऑफ को-स्टिमुलेटरी लिगेंड्स सच एज CD40 एंड B7 मॉलिक्यूल्स और एक्ट इनडायरेक्टली बाय इंडक्शन ऑफ रेगुलेटरी टी सेल्स. मतलब डेंड्रिटिक सेल्स या तो इंडक्शन करा देंगे रेगुलेटरी टी सेल्स का, ट्रैग सेल्स का, ठीक है, या तो ये क्या करेंगे जो कि फर्दर अपना इंटर साइटोकाइनेंस रिलीज करते हैं लाइक इंटरल्यूकिन 10 एंड ट्रांसफॉर्मिंग ग्रोथ फैक्टर जिसके कारण डाउन रेगुलेट हो जाते हैं सेल्फ रिएक्टिव टी सेल्स. या तो ये क्या करते हैं और डाउन रेगुलेट कर देते हैं एक्सप्रेशन ऑफ मॉलिक्यूल्स ऑफ को स्टिमुलेटरी लिगेंड्स सच एज CD40 एंड B7 मॉलिक्यूल्स, उनको ही डाउन रेगुलेट कर देते हैं एक्सप्रेशन ना उस मॉलिक्यूल्स का तो जिसके कारण क्या होता है कि we can't recognize the antigen, so there occurs the peripheral tolerance. Last but not the least, so this is the sequestration of the self antigen. अब क्या होता है कि certain antigens जो होते हैं they are normally present, they are normally not they are normally not presented to the immune system. uh, uh, they can evade the immune recognition by uh, and certain कुछ सेल्फ एंटीजेंस होते हैं वो क्या करते हैं इवेड कर देते हैं इम्यून रिकग्निशन को बाय सीक्वेस्ट्रेशन इन द इम्यूनोलॉजिकली प्रिविलेज्ड जो कि सेल्स होते हैं। तो इम्यूनोलॉजिकली प्रिविलेज साइट्स कौन कौन सी होती है जहाँ पर ये जाके इनकी सीक्वेस्ट्रेशन हो जाती है जिसके कारण ये इम्यून सिस्टम में विजिबल ही नहीं होते। दीज आर कॉर्नियल प्रोटीन्स, टेस्टिकुलर एंटीजेंस एंड एंटीजेंस फ्रॉम द ब्रेन। So by this method, the peripheral tolerance is basically seen in our immune system and our body recognizes the self from the non-self. So today we are going to discuss now the mechanism of autoimmunity. So it results due to the breakdown of one or more of the mechanism of immunological tolerance, which can be central or peripheral in nature. So first is the antigenic alterations. So cells or the tissues may undergo antigenic alterations such as it may as a result of physical, chemical or biological influences, and hence it is named as new antigens. So new antigens can be produced by any of the following methods, by any of the following agents, and it results due to the alteration in the antigens basically. So physical agents, which includes irradiation, photosensitivity and cold allergy, viruses, intracellular pathogens, bacterial enzymes, for example, neuroimmunidase and many bacteria, which acts on RBCs releasing T antigens. The other causes may be mutation, all results into the new antigens, and there is an alteration in the self antigen. Second is the release of the sequestered antigens. For example, the self antigens which is present in the closed system, which are never or which are not accessible to the immune apparatus. For example, eye lens protein, sperm protein, antigen in the brain, when they come in the when they appear, they results into the autoimmunity. Another cause is the cross-reacting antigens. In this, there is a similarity between the foreign and the self antigen. So it is basically present on the organ, and on the organ there is a specific antigens which is present in the body. Organ-specific antigens is present in the body. And hence the injection of an heterologous organ-specific antigens will induce an immune response. For example, the complication of neural vaccines and rabies, cross-reaction between the human and the sheep brain antigens is one of the most classical example for this thing. The one of the second most common is the streptococcal M proteins, which cross-react with the cardiac muscles. And there is one more, for example, there is a nephrogenic strains of streptococci which possesses the antigens which are found in the renal glomeruli. So instead of identifying itself, instead of identifying and instead of showing immunotolerance to the self antigens due to the cross-reacting antigens, the autoantibodies or the antibodies does not recognize between the self and the non-self, resulting in autoimmunity. Next one is the molecular mimicry. There is a presence of epitopes with identical peptide sequences in some infective organisms and the self antigens. The same as we have explained it before. For example, the arthrogenic Shigella flexneri and HLA-V27, they are having the molecular mimicry, and the Mycobacterium tuberculosis and the joint membranes, whereas the Coccus virus and the myocardium, they have the presence of epitopes with identical peptide sequences on their surfaces. The other thing which results in the development of an autoimmunity is polyclonal B cell activation. In this, there are certain stimuli which non-specifically turn on multiple B cell clones. For example, these stimulus can be chemicals, 2-mercaptoethanol, bacterial products, enzymes, antibiotics, infections with mycoplasma, viruses, and parasites. And these polyclonal antibodies are basically IgM in nature. Next one is the forbidden clones. In this, there is a breakdown of immunological hemostasis, which may lead to the cessation, that means cessation of tolerance. When there is a cessation of tolerance due to the breakdown of immunological hemostasis, there is the emergence of forbidden clones of immunocompetent cells. That means the immunocompetent cells, they will not, immunocompetent cells which are capable of mounting an immune, they will be capable of, since there is a cessation of tolerance, तो क्या होता है कि जो immunocompetent cells है वो immune response generate करते हैं और they mount the immune response against the self antigens, resulting in again the autoimmunity. So what is the causative factor for this thing? There is a breakdown of immunological hemostasis. The other factors which include are altered T and B cell function. The T helper cells facilitate the B cell response to antigens, while T suppressor cells inhibit antibody production by B cells. So optimal antibody response depends on the balanced activity of T helper cells and T suppressor cells. Enhanced helper T cells and decreased suppressor T cells functions results in autoimmunity. There may be defect in pathogenic stem cell development and the macrophages, and there can be defect in the idiotype anti-idiotype network. All these factors results in ultimately the autoimmunity. That means the person immune system does not recognize self from non-self, and hence developing the various autoimmune disorders. So now we are going to discuss the mechanisms of tissue damage in autoimmune diseases. The types of autoimmune reactions, it can be autoantibodies which develop against self components. The second is antigen antibody immune complexes which deposit in the organs. The other mechanism of tissue damage include autoreactive T cells against self components. And last but not the least is combination of the above